in this last part, we are going to do a little bit of introduction into genomics, just a little bit, and population genetics, just the basis. So when we do genetics, we can check the genes one by one. We can do it in the high definition, or you can check them all together. So in a population, let's pick two people randomly, Ali and Ahmed, and let's compare their chromosomes. In this case, we are looking at their chromosome 2, and we realize that Ali and Ahmed are very different on chromosome 2, not because they are strangers or something, but because normally every individual is different from each other in 1 in 1,000. That's very normal. So they are different with these gray spots that I have drawn. So they can be small differences. Like in this case, this is the 1 nucleotide difference, or in this case, the difference can be bigger, okay? Maybe much larger. So some of them are single nucleotide differences, and some of the differences are larger. Maybe it is missing part of the chromosome. Maybe it has a large deletion here and not affected. Or maybe it has a large insertion. Or it can have a copy number variation, because we know that like 95% of our genome are non-functional. It is full of repeating and non-repeating elements, and in these situations, in these repeating and non-repeating elements, some of the repeat numbers change from person to person, again, which is very normal. Let's say in this case, for example, Ali has 3 repeats, and let's say it's a 10 nucleotide long short repeat, and Ahmed has 300 repeats. So it makes these two people different at the level of genetics. But because it is not controlling the gene expression profile at all, we don't care. So these two people will never know that they are different at this level, at this for this position, because they never have to know. Because we know that only the minority of the genome is coding. Let's mark the genes here. Let's say there's a gene here, another gene here, another gene here, and here. Maybe it's close to the terminus. Let's call them gene A, B, C, and D. Now, most of the differences that we observe between people, they're going to lay out of the genes. Here, actually, I'm more optimistic. So there are 4 differences. Each gene has a difference. This is not usually what we see. Majority of the differences are in the intergenic region, because this variation in the intergenic region, it doesn't affect the phenotype. So if there is a variation in the non-coding intergenic region, so it doesn't change gene expression profile, and because it doesn't change the gene expression profile, it doesn't change fitness. And because it doesn't change fitness, it has no effect on the organism, and it can easily stay there. Nobody will notice, nobody will care. Okay, now let's say for this position, Ali has thymine and Ahmed has cytosine. Now you wonder which one is normal and which one is abnormal, because as human we like to call things normal and abnormal. By the way, let's make them a different color. Say it is T here and C here, and we think that one of them must be normal and the other one must be mutant or abnormal. So we cannot decide just by comparing Ali and Ahmed. So what we do instead, and we go check the rest of the population. In the rest of the population, we see that like 99.99% has thymine, and 0.01% has cytosine. In this case, Ali is normal and Ahmed is a mutant. Or maybe you check the entire population of that city and you never see anybody who has cytosine at that point. Ahmed is the only one. Then you would say that, okay, Ali is normal and Ahmed is abnormal. By the way, normal doesn't mean good, normal doesn't mean beneficial. It just means more common in your life too, when something is normal just because more people have it. So, in this case, we just neglect Ahmed is a mutant. Many people don't have it, but there can be other cases too. Maybe in the population, 60% have thymine, 40% have cytosine. In this case, I wouldn't call anybody normal. Well, they are both normal because half the population has thymine, half the population has cytosine. So then I would call this region polymorphic. Okay. Maybe for the neighboring nucleotide, everybody has guanine here. So when you check the entire population, the next nucleotide is guanine in everyone, except the rare mutants. But when you check this position, it is half and half. There is no normal. So this region is now called polymorphic. And if you check it in a single nucleotide level, you can call it a single nucleotide polymorphism. It is not pronounced as SNP, it is pronounced as SNP. So get the nomenclature right. This is, well, SNP is kind of single nucleotide variation. Okay? Variation means it can be mutation, it can be less variable. For something to call polymorphic, for a region to call it polymorphic, the frequency of the nucleotide will be high enough, should be high enough. Otherwise, we just call it a variation or a mutant. To call it polymorphic, it must be high enough. Let's say if you see that nucleotide in 2% of the population, then it's polymorphic. You can call it a single nucleotide polymorphism. Now, single nucleotide polymorphisms are very useful. Okay, last time one of you asked a question. SNPs are very useful. Useful for whom? I mean, it's obviously not useful for Ali and not useful for Ahmed because, anyway, this nucleotide lies in the intergenic sequence. It doesn't affect the gene expression. It doesn't affect the fitness. So they are useful for us if you're going to do genetics, genomics, bioinformatics, or even medicine. SNPs are extremely useful because they allow us to follow people, they allow us to identify things. Yeah. In this case, for example, they identified 3 SNPs. So in this case, SNP1 exists 50-50 in the small population. SNP2 exists in 1 in 4, and SNP3 also exists in 1 in 4. Okay. And when you check the chromosomes, yes, regularly we see them. So frequently we have them. So when the human genome was sequenced for the first time, even before that, even before the human genome was sequenced, people identified some SNPs so that they can follow the genes, they follow the location and segregation of the genes, but there were just few hundred. As the human genome sequencing is human genome sequence is published, and as they continue sequencing different people, they identify more and more SNPs. Currently, there are tens of thousands of SNPs that you can work on. So it's like a very large data that people are trying to get meaningful results of. For a bioinformatician, it's heaven. So at one point, I need to define something else, haplotype block. Okay, let's think about Ali. Now, Ali has all these mutations, and after that, Ali gets married, have some children, and of course, when he is having children, he has meiosis. This is how we have children. And in the meiosis, something happens very well, crossing over. If there is no crossing over happening here, which is quite unlikely, Ali is going to inherit this chromosome 2 to his children as it is with all these SNPs. But if there is any crossing over happening, let's say a crossing over happened here, another crossing over happened here. This is going to break the chromosome, and in the next generation, this is what we are going to see. Alice, let's say son, will not have these SNPs. These SNPs will not be inherited. And in one single chromosome, by the way, he was diploid like every human. And in the other chromosome, he had other SNPs, or maybe no SNPs at all. The SNPs that he is going to inherit to the next generation are these. Okay. This is called a haplotype block. Because these SNPs were inherited together in a single generation. This is Ali, maybe this is his son Hassan. Okay, and imagine that Hassan is also having a child. Again, he's having crossing overs. Again, he has a crossing over here, but because this part, the SNPs there is gone, we don't care. And then he has another crossing over here, which happens totally randomly. The haplotype block will get smaller from here to here. Okay, so each time there is meiosis, because of the crossing over events, the haplotype blocks get smaller and smaller. Haplotype block is a sequence of chromosome where the original original collection of SNPs are kept. Okay, so as they have children, they are going to have smaller, smaller, and smaller haplotype blocks. So why is it important? Okay, Hassan is the son. Maybe he has a sister called Ayşe, and Ayşe has another son, Yiğit. So Yiğit and this guy, let's call them Murat, are cousins. Yiğit and Murat, they're going to share some DNA, and the DNA they have is going to have some common haplotype blocks. So even if Yiğit and Murat, they don't know each other at all, even if they grow up after a war, that they lost all the family, just by checking their genes, just by checking their SNPs, you can understand they are cousins, or at least they are relatives, because they're going to have very large haplotype blocks. And in the meantime, they have a neighbor. This neighbor is called İdil, and she actually came from a different city. When you compare İdil, Murat, and Yiğit, you wouldn't see any haplotype block between Yiğit and İdil, because İdil is quite a stranger. She was born in a different population and then migrated and came in. This is how we use the haplotype blocks. Okay, in this example that I give, I have just shown like 3 generations, but normally we count like 10, 20, 50, 100, 200, 300 generations. Okay, because there are like thousands of SNPs in every chromosome. We check the original compositions of the SNPs, how large they are. And one of the biggest applications of SNPs is the human migration paths. We know that the Homo sapiens, they first evolved here, and then from here they migrated out of Africa several times. And as they migrate, of course, they had children. And of course, each time they had children, their genome was changing, the SNPs were breaking and recombining, and their haplotype blocks were getting smaller and smaller. So if you check a population here, okay, Siberia or Yakut, I'm not sure. If you compare 2 people here, 2 individuals here, you're going to realize that their haplotype blocks are very large because they are a uniform population. But if you compare this with this, which diverged long time ago, their haplotype blocks will be really smaller. So if you compare one person here and one person here. And this quite makes sense, because if you compare people from 2 different regions, yes, they are going to be different. So what makes people different is the mutations they accumulate through time, and we usually assess these mutations through the SNPs. SNPs tells us a lot. Okay, so as two SNPs are together in the same order, in the same organism, and there is another SNPs in their neighbor, you can say that, okay, the people who live in, not Alaska, I'm sorry, Yakut land, they have this combination of SNPs. So this is specific to these people. This is how people, there are some companies and popular videos here. You are like 20% Turkish and 50% British and 10% African, etc. They mostly depend on these haplotype blocks. There are some haplogroups that you can easily find on internet and read, but be careful, some of these haplogroups give some specific cities or something. That's impossible. Genetically it is not calculatable, so they are concluding too much from a little data, so I wouldn't consider them correct, not ethically, but simply they are lying. And well, I should have a couple of words about variation too. So this is the old continent Africa. So Africa is the source of human genetic variation because humans evolved there. 85% of the entire variation of the human genome is still here. And 15% of the human genetic variation, they stay in the non-African population. So if you're going to ever do genetics, you should divide people into two. I usually divide people into two, good people, bad people. But as a geneticist, you should divide them into two, non-African and African. And non-African contains everything. The East Asian, the European, the, the islands here, the American nation, locals, all of them, they are included in this 15% genetic diversity. And if you're going to do statistical test, you're going to realize that the statistical test you have to use for the African population is different from the statistical test from the non-African population, because these guys are more or less uniform, while here you're talking about a great variation. And because of this variation, you should use other parameters to get a meaningful data out of it. Okay, so if you're going to, for example, compare the haploblocks, of course it's much easier if you check the American Indians. The American Indians are, they have a clear-cut migration path, so they have large haplogroups. You can easily identify them. But what if you compare a village here and another village here? They can be product of two different migrations. They are going to have giant difference between them. You will not be able to identify any haplotype block between them at all. Okay, this is one thing that you need to know about genetics. The second thing is, when people migrated out of Africa, when they were going east, here they mated with Denisovans. It's a kind of humanoid. It's very similar to Homo sapiens, but it's not Homo sapiens. We are still not sure. We still didn't classify it. And these Denisovans also contributed the genotype of these guys living in these islands and also in New Zealand and Australia up to 8%. And the people that migrated to Europe and Asia too, they mated with Neanderthals. Well, we call them Homo neanderthalis because they were found in the Neanderthal valley, and here they were found in the Denisova, which is a Turkish name, which was a cave. They were named after the caves and valleys they were found. And we can say that these Homo sapiens, yes, they have been away from Neanderthals and Denisovans for a long time. So they were genetically distinct, but the distinction between these three humanoid groups were not large enough to make them different species. Because, the speciation is a slow process. When a population is divided into two, they start to differentiate, they start to accumulate mutations. If enough time passes, if they accumulate enough mutations at one point, they cannot breed together, so you kill them two different species. Like, for example, elephant and horse. They are two different species because there has been long time after they diverged. But horse and donkey, they can still mate. So can we call them two different species? Yes, still we can, because their child, mule, is sterile. And Neanderthals, Denisovans, and Homo sapiens, we are not sure if we can call them different species because we know that they can mate. And after they mate, they have productive progeny. They can have grandchildren. So I think they're still in the same species. And it was very useful for the humans because Denisovans have been here much before than the Homo sapiens came, and Neanderthals have been living in the cold climate of Europe. And these guys, they were adapted to local parasites. Parasites are one of the big drives in evolution. It drives the immune system. It induces the natural selection. And because Homo sapiens mate with these people, they have taken the alleles which gave them immunity, or here, for example, the cold climate in Europe, the resistance to this, or the light skin, which will allow them to have the vitamin D enough. Okay, and of course, when you check the haplotype blocks of these guys, you would see some specific haplotype blocks that are specific to these people who is living in this area. Okay. This is not the only place that we use them. Sometimes we use SNPs as markers. Of course, when I say marker, I don't mean that, okay, I'm going to mark this person Asian and I'm going to mark this person European. No. These are markers of diseases. For example, imagine that this is these are the genomes of the healthy controls, okay, and these are the genomes of the, let's say, people with breast cancer. Okay. Well, you see that they have different genomes, and these are the SNPs. Okay, so just by checking the SNP, you can say that whoever have this blue SNP would be healthy, and whoever has this red SNP would be sick. Okay, you can easily say that even if this SNP is not causative, even if this SNP doesn't cause the disease itself, because it is located close to the gene which causes it, they are going to be inherited together, or as I say here, they are going to be linked, just like B and A. Maybe B is the causative gene, maybe A is the SNP marker. Just because they are very close, whenever you see SNP B, you would also understand that the disease-causing allele would be there. Okay? And the rest of the genome, you don't care. And what people do, they get genetic information from different places, different individuals. They compare different populations, and then they decide, okay, the people who has this SNP, they have a high tendency for breast cancer. The people who have this SNP, they have high tendency for lung cancer. The people who have this SNP, they will be resistant to coronavirus infection. Lots of statistical analysis is going in a GWAS, which is a genome-wide association study. It's a large data set, and then you get few meaningful SNPs, okay, which goes beyond this threshold. So all for genetics, of course, it is very basic, it is very fundamental, but once you get the basis, you can read it yourself.